It's a real pleasure to introduce Fred Moore to you this morning. Fred and I have been friends for many, many years, and looking at his career, Fred graduated from the University of Pittsburgh, did his training at University of Colorado, Denver General, where he stayed on the faculty for many years before he moved to UT Houston, where he was the Director of Trauma, Chief of the Division of Trauma, Acute Care Surgery, and Vice Chairman for the Department of Surgery. And several years ago, Fred moved to the Methodist Hospital just across the street from UT Houston, where he is the Director for Acute Care Surgery and the Surgical Intensive Care Unit. Fred is a true example of what a triple threat is. Fred has made a number of contributions to the surgical literature and to the trauma in particular. I think it's hard to list all the contributions that Fred has made, but I think if you read anything related to multiple organ failure in surgery in general, not just in trauma, but in surgery, you're going to find Fred Moore and his brother Gene Moore's name on it. Fred was one of the first individuals with his brother and Dr. Ben Eisman to study multiple organ failure and describe the two-hit theory. That first, there is the first hit, then you go through a period of ischemia and reperfusion, you're resuscitated, then you develop multiple organ failure because of the tissue injury related to ischemia and reperfusion. Fred's a basic scientist, he's a clinical scientist, he's a great educator and a very, very talented surgeon. Fred was past president of the Western Trauma Association. He has served on a number of committees in any and every major surgical association in this country and internationally. And he is the president-elect the president-elect of the Southwestern Surgical Association, and he will treat us this morning with a preview of his presidential address to the Southwestern Surgical Congress. So Fred, thanks for coming.
Thank you, Raul, and I want to thank Mark for the wonderful dinner we had last night. It's always interesting to go to different places and see where your friends work and see the environments that they develop, and certainly the trauma center that you have is a real jewel in the trauma community. So my topic today, the title is Imagination Trumps Knowledge. And this is, I'm going to focus on the use of cartoons as a strategy in translational research. Raul mentioned Denver General Hospital. I went there as a fourth-year medical student, and we called it DG. And at the time, it was a prototype knife and gun club. And it was an inner-city hospital, had a very advanced EMS system, and the activities that went on there really are the reason I'm standing here today. Now, the chief of surgery there was a guy named Ben Eisman, and Ben was one of these people that, you're a medical student, you just love walking around with him because he'd tell you things that you'd never heard of, and he'd tell it to you in a very interesting way. And he kept talking about multiple organ failure. So this is 1978, and I'm saying, well, I've never heard of that. So when I went and looked it up, it turned out that he coined the term in 1977 to describe a new syndrome that was occurring in the ICUs at Denver General. And basically, he observed that patients were surviving single organ failure to get multiple organ failure. And he and other thought leaders at the time believed that this multiple organ failure was the result of uncontrolled infection. So that sort of set the stage for how we approached this. And this confusing and important topic really became DG's research focus. Now, I was just talking to Mark about nutritional support teams, and I, as a resident, got involved with this nutritional support team and began to do things with them as a resident. And at the time, we really were hyped up about nutrition, and that's because total parenteral nutrition was becoming available, and we had to have these teams so we could administer it safely and then study whether it was effective. So our team had this little cartoon, and we were trying to explain how nutrition and multiple organ failure were linked, and it turns out that trauma patients, when they're really ill, they get hypermetabolic, and because they're hypermetabolic, they develop acute protein malnutrition, and the body mobilizes substrates. There's wasting of skeletal muscles, wasting of visceral proteins, there's organ dysfunction, and most notably, the immune response becomes impaired. You get infections, and you get multiple organ failure. So that all made sense, and our hypothesis was, well, if we could just get in there quickly and feed the patient, that we would prevent this from occurring. Now, the first study we published was comparing early total enteral nutrition versus delayed TPN, and we demonstrated that there was reduced infection, so it kind of fit the hypothesis. The next study, however, was comparing early TEN, total enteral nutrition, versus TPN, and we showed that early enteral nutrition was associated with decreased infections. Well, that didn't quite fit the hypothesis because both groups got good nutrition. And we really couldn't explain why enteral nutrition, was it because enteral nutrition was good, or was it because TPN was bad? Now, my oldest brother was sort of teaching me how to do research, and he said, well, we got to draw a cartoon. And so this is the cartoon we drew, and we said the gut was the starter, the liver was the motor, you had bacteria and endotoxin inside the gut, you had shock, you had delayed enteral feeding, and you had bacteria leaking up through the portal vein, activating Kupffer cells, and a lot of mediators. And, you know, why would we draw a cartoon? Well, the purpose of drawing a cartoon is to develop a win-win hypothesis, something that you could test, and whether it was true or false, you would have added something to our knowledge. So our hypothesis was that bacterial translocation via the portal vein was a driving mechanism in this multiple organ failure syndrome. Well, about this time, Dr. Harkin had become the chairperson at the University of Colorado, and as he was surveying the people that work for him, he recognized that the Denver General group was doing some fine research, and he came down to us and said, we're going to crank this up a little bit, and we're going to develop a trauma research center. And this trauma research center is going to focus on people. So, favorite quote of his, if it doesn't have anything to do with Mrs. O'Flaherty in the ICU, then we don't want to study it. And so the ICU at DG was made the clinical core, and I was made the director of the clinical core. Now, our first study with Dr. Harkin's enthusiastic support was this one, where we tested the hypothesis about bacterial translocation in a portal vein. We took 20 high-risk trauma patients, and at laparotomy, we dilated up the umbilical vein, and we stuck a catheter in the portal vein. And we left it there for 5 days. And we sampled serial samples looking for all sorts of mediators, and 7 of them actually went on to develop MOF. Now, to our total surprise, there was no endotoxin or bacteria in the portal vein. So it's a totally negative study. But yet this was one of the most quoted papers in the 90s out of that journal, because so many people were convinced that bacterial translocation was the cause of all ailments. So since that didn't work, I had to go back to the drawing board, and my role as the head of the clinical core was to draw little cartoons and take them to the research meeting that we held every Friday at 6 a.m. And then I would present these things, and they would normally laugh at them or not really like them. But this one they sort of liked, and it was that you have a first hit, which is a combination of shock and tissue injury. You develop a systemic inflammatory response, and then some people you really can't resuscitate, and they kind of just die early. And then there's a group of people that come out of this who appear vulnerable, and other people appear to be protected. So an example would be somebody who aspirates, or somebody who has their femur rotted at the wrong time. That can function as a second hit, and you activate the system and they get MOF. Now, we kind of modified this cartoon and we wrote a trauma center grant, and the title was Trauma Primes for MOF. And in the lab, we studied priming and we studied preconditioning, two different things. This is harm, this is protection. And then we described a thing called the two-hit model. And we worked at that in the laboratory, just, proving that that actually is a valid concept. Now, because we had this neat cartoon, Dr. Harkin got excited and sent his PhD down to talk to us. And Renato Paghetti was a Brazilian from São Paulo, and he came and joined our research group. And together they developed a rodent model. And this review article nicely summarizes, and here's another cartoon. We have activation of gut PLA2. We have neutrophils sequester in the gut, and that's MPO. Then we have circulating neutrophil priming. We have PMNs go into the lung, and we get lung leak. And then we found that these priming mediators were actually lipids in the mesenteric lymph. Now, priming of circulating neutrophils became the focus of our clinical and laboratory studies, and there was an SUS meeting, and I had to get up and explain to everybody what we were talking about. And so this was another cartoon where we showed that gut ischemia reperfusion causes it to become the priming bed where quiescent neutrophils get primed and activated, or primed and increased adhesion receptor expression. They didn't get activated and sequester in the end organs. And if you're going to study this clinically, then you want to study this phenomenon by doing blood sampling. We recruited a general surgeon from the UK and a pediatric hematologist, and we set out to write this review article and sort of talking about what priming meant and how we might study that. And then we went and did a series of prospective focused observational studies where we confirmed that neutrophil priming and activation occurs, neutrophil sequester to cause organ failure, platelet activating factor and IL-8 are the mediators that are involved with this priming. And so this was a proof of concept, moving from a cartoon to the laboratory to the bedside. This lady, Angela Sawaya, joined us from São Paulo, and she was an internist, and she wanted to get a PhD in statistics, and she was interested in predicting things, and so we set off to develop the Denver MOF database. Now, after 4 years or so, we had collected enough data that we could actually write a paper and talk about what is the current epidemiology of multiple organ failure. And there are really two forms. There's an early form that has nothing to do with infection, and there's a late form that's probably induced by infection. So this is the cartoon that we drew in that paper. We said that trauma sets up a systemic inflammatory response, and if it's severe, you get early MOF. And then as time goes on, the body down-regulates the inflammatory response. You get severe immunosuppression, infections, and you get late MOF. Now, we were talking about the innate immune response. That's neutrophils. That's what we studied. They're very important in this cascade. But in the same year, a gentleman named Roger Bone described something he called CARS, the compensatory anti-inflammatory response syndrome. And he was talking about the adaptive immune response. Well, the CARS kind of fit nicely with SIRS, and so we put that in our cartoon, and that's fair. You're allowed to do that when you're making these cartoons. And it turns out that this fits with our SMAO model. You have shock, gut ischemia, reperfusion, pro-inflammatory mediators. So that all kind of fit, and therefore we were happy with this cartoon. Now, I went to Herman Hospital in 1996 and worked with the surgery department at UT to develop a research program. And these were the attending surgeons I inherited. And like most trauma surgeons, they worked hard and had lots of opinions. And when I told them we were going to do research, they weren't quite sure if that's what they wanted to do. And I said, well, you got to look at this cartoon, because this really makes a lot of sense, and we could make a contribution here. Because if you look at these risk factors, shock is a variable that we can treat. And so we said this is going to be the focus of our clinical research. We're going to study shock resuscitation. So I developed this bedside algorithm when at Denver General, our group was very interested in this phenomenon and developed algorithms to drive resuscitation. I had been working with these two gentlemen doing computerized decision support for mechanical ventilation of ARDS. And when we finished this trial, I said, well, if you can actually use a computer to run a ventilator in somebody who's dying of ARDS, why can't we do that with shock resuscitation? And so we started the process of making these algorithms. And this is what I thought we were going to do. There would be me, I'd get a bunch of people, and we got all those papers in my office, and we'd put them through there, the knowledge engine, and we'd make algorithms, we'd give it to Matt, and then it would be all over. But it turned out not to be that simple, because the first step is you have to make paper protocols, and you need to test them at bedside. And you need to get nurse facilitators, you need to work on the logic and say, well, wait a second, that's working. And that's then you can proceed on and get the programmer and start making sure that the program works on the computer, teaching patients. And it's really an iterative process. You evaluate and test every step. It's never done. You're always monitoring and refining it. So it became the standard of care, probably about 1999. Bedside real time. We had a database. We collected data. And the thing we were trying to do is standardize care. And then with ongoing analysis, we refined the protocol. And we collected the epidemiology outcomes of abdominal compartment syndrome, multiple organ failure, and death. Now, UT had a trauma center grant, and Dr. Frank Moody was the PI, and the other major player was Norm Weisbrot, who was a physiologist. And they were interested in gut inflammation and ileus. And so I wanted to see how I could work into that. This was the paper that they were studying, that they had published right when I got there, looking at the role of inducible nitric oxide synthase in sepsis-induced ileus. They used LPS as a way to induce sepsis. And they had a really cool intestinal transit model where you take a catheter and you surgically put it into the duodenum. You let the animal awaken, and then in a non-anesthetized, unrestrained state, you can study intestinal transit. And the way you do this is you do your experimental insult or sham. You wait at least 5 hours. You then inject a fluorescent tracer, wait 30 minutes, divide the bowel into 10 equal parts, and then you remove the tracer, and you can figure out where the tracer is and calculate what's called the mean geometric center. And this, for me, was a functional outcome that would be consistent with the ability to feed the GI tract. Now, I call Hytham my diamond in the rough, because about this time I was very interested in getting our lab going, and I kept telling Dr. Andrasi I need to start a lab. And so one day Hytham shows up, and he is from San Antonio. He's a second-year surgery resident. His wife wants to move back to Houston, and he wants to move back with her. Well, there's no jobs in Houston, so could he work in the lab? And I said, well, of course, that'd be great. And we quickly figured out that we just take this Denver SMAO rodent model, we'd measure intestinal transit, characterize gut inflammation, and use all these INOS blockers that the other investigators were doing. And then we began to have a discussion with these people, and after a couple years, Hytham was prepared to write a review article. And each one of these co-authors were people that had R01 grants. And what we were trying to do is to get everybody on the same page and come up with a story of life to explain how we all work together. And this is the cartoon that we came up with. This is after about 10 renditions, but it makes a lot of sense. You have shock, gut ischemia, resuscitation, reperfusion. You can do that, but with reperfusion and resuscitation fluids, you get some gut dysfunction. If you do a laparotomy on somebody, manipulate the bowel, it sets up an inflammatory response. ICU therapies, everything we do is bad for the gut. We give H2 blockers, take away gastric acidity. We give morphine, that causes an ileus. We give a bunch of antibiotics that kills off the normal flora. Then we don't feed the patient. And the end result is you get a variety of gut dysfunctions, and over time, the gut becomes heavily colonized, and it's got an ileus, and you get aspiration, translocation, infections, toxins, late MOF, and then this sepsis feeds back and causes additional gut dysfunctions. And thus we proposed that the gut was the instigator and victim of this response, and that we were going to study this. Our global hypothesis was that novel therapeutic interventions can modulate gut inflammation, injury, and dysfunction to improve outcome. We proposed 5 laboratory projects. One was a renewal of Dr. Moody and Weisbrot, and then 4 new projects. We had 3 interactive cores, human subjects, molecular, and administrative. And the first thing that I learned long ago when working in Denver, you got to have a research partner. Surgeons get very busy. You got to have somebody who you can sort of, when I'm not around, you'll be around. And that person turned out to be David Mercer. He was a professor of surgery, chief of surgery at LBJ, and his interest was the stomach's response to stress. And through a number of discussions and doing some research with his investigators, we came up with this cartoon. He's going to use SMAO, ischemia reperfusion, or LPS. We're going to look at transcriptional factors, pro and anti-inflammatory genes. We're going to look at the inflammatory response and the resulting mucosal injury, luminal alkalization, stasis, and impaired emptying. And these are the modulators that we were proposing to test. But as we got closer to submitting the grant, we found that the sedative ketamine was extremely protective in these models, and he started investigating how ketamine does that. In fact, the Army has done a number of subsequent studies showing that it's very protective in the setting of hemorrhagic shock. Now, the other thing that is important is to somehow attract the superstar researchers, and Bruce Combe was one of those. He was the chairman of medicine, renal consultant in our ICU, but his research interest was the molecular biology of renal ischemia reperfusion. I sat down with him one day and said, well, that's a nice subject, but why don't you study something important like gut ischemia reperfusion? And so I sent him Hytham, and he had something called alpha melanocyte stimulating hormone, and this is a protective hormone. And we drew this little cartoon. Gut ischemia reperfusion affects stress kinases, transcriptional factors, pro and anti-inflammatory genes. And Hytham showed that alpha MSH decreases NF-kappa B activation. Bashir showed that intra-ischemic hypothermia protects by inducing hemoxynase 1. And then Stacy Moore developed a preconditioning model and did a microarray gene analysis to look for novel genes. Now you can see why Dr. Combe was attracted to us. I gave him a series of 3 surgery residents that did a tremendous amount of research for him, and he got a number of publications, and so did we. So about this time, we're well into this computerized resuscitation, and if you were to ask my group of attendings who were initially skeptical, they said, well, this is pretty good. In the field, EDOR, we use advanced trauma life support. We do a lot of damage control surgery. Then we take those patients to the ICU early and we optimize systemic inflammation. And people who previously just died seem to be living. But something ugly had appeared. called the abdominal compartment syndrome. And this particular entity had arisen across the United States, across the world, and people were kind of puzzled about it, and we were well positioned to study it. Zsolt Balog was a visiting Hungarian trauma surgeon who was working with my brother in Denver, and he'd never really seen this thing, and he got very interested, and I said, well, come down and look at our database. I don't have time to analyze all this data, but you should be able to answer some important questions. So he did a series of 4 database analyses, and I'll quickly go through them. This was just a simple description of secondary abdominal compartment syndrome. This was looking at the epidemiology of primary and secondary abdominal compartment syndrome, showing that we could predict it within 3 hours of the patients in the ICU, that people who got it were going to get multiple organ failure. This one was showing that these people who had impending abdominal compartment syndrome really didn't like the resuscitation protocol. And the 4th was, as we refined our protocol, we decreased the oxygen delivery goal. And when we did that, when we did less vigorous resuscitation, the abdominal compartment syndrome decreased, but did not disappear. We wrote this invited opinion, and the reviewers were really very upset with us. And they didn't really understand the intent of it. The intent of it was to set up 2 laboratory projects that were going to go into our center grant. And so the first proposed laboratory study was for Chuck Cox, and we wanted to look at how standard resuscitation fluid causes gut edema and dysfunction. And the cartoon we came up with is here. And Chuck has done a number of very novel experiments looking at how gut edema in and of itself affects inflammation and causes dysfunction, notably ileus. Now, my project was to look at alternative resuscitations and how it affected gut ischemia reperfusion inflammation. Bashir had done some very interesting work, preliminary data showing that hypertonic saline protects in this model in part by inducing heme oxidase 1. So we had some nice preliminary data. And by now we had characterized this model, and we knew the inflammatory markers. We had a good handle on the timeline of the injury and edema. We had a functional outcome. And what we were proposing is that we kind of know how this works. We've got gut ischemia reperfusion, activates AP1, NF-kappa B, turns on INOS. We get PMN influx, we get injury. And what we had discovered was that any insult you do immediately turns on protective mechanisms that try to reverse the injury. And the 2 that we identified were PPAR and heme oxidase 1. So the proposal was that we were going to look at how alternative resuscitation strategies could turn on this protective anti-inflammation and allow the injury not to occur. So as we got our program project together, we came up with this cartoon, and we sent our grant in. Initially, it really didn't get funded, it didn't get a fundable score, but it was a score that they wanted to come and see us. So they did a site visit, and they really didn't believe this. They just thought this was something, we figured out the night before and put it in the grant. But when they came, they actually saw that this was true. And that I was, while working in the laboratory, knew a lot about clinical research. And similarly, while Dr. Mercer knows something about clinical research, he knows a lot about laboratory research. And we had organized all these people around this common theme. Now I just want to quickly show you how a young investigator can benefit from this. Rosemary Kozar came to me, very well trained. She was a trauma surgeon from Hahnemann, and she came and said, I want to be a trauma surgeon. My husband's going to get a job here. And I said, well, I'd be happy to have you, but you've got to do a critical care fellowship, because I really don't have a use for a trauma surgeon who doesn't have critical care boards. And then I said, Rosemary, it's not that hard being a critical care fellow, and while you're there, you can figure out what you want to do research on. And so we sat down and we kind of reviewed the cartoon, and she kind of understood it. And she was going to go in the ICU, so we said, why don't you pick an ICU therapy? And she liked early enteral nutrition. And we had developed an early enteral nutrition protocol in the ICU. And this protocol had a way in which we advanced formula and we monitored for intolerance, or we managed it. And so there was a protocol that was pretty good. And during her critical care fellowship, she exported this to 3 other trauma centers and showed that if you use this protocol, almost all trauma patients can be fed via the enteral route. Now, the reason we developed this protocol was because when I first got to UT, they had a new tube that they were extruding a nasojejunal tube way out into the jejunum. And if you didn't monitor that feeding, you could kill the bowel. And I really hadn't seen too many cases of that. And so we wrote up this series, and again, based upon what I was taught in Denver, this is a confusing problem, so let's draw a cartoon. And there's really 3 different ways this could occur: metabolic stress, dysmotility, or bacterial overload. And Rosemary said she wanted to study this. You know, how could nutrients cause metabolic stress and cause microvascular ischemia and cause the bowel to die? So we began to investigate this, and it turns out that certain nutrients like glucose, fructose, and glutamine, and alanine and arginine, they are all absorbed using ATP with the exception of fructose. And then once they get into the cell, glucose, fructose, and glutamine can produce ATP where alanine and arginine cannot. So that would mean that alanine and arginine might be hazardous to the intestinal mucosa in the setting of hypoperfusion. So she developed a jejunal sac model using 45 minutes of SMAO, 30 minutes of reperfusion, filled the sacs with these agents. And this is one endpoint she looked at, deconvolutional microscopy, looking at the villus. So glucose seems to protect, fructose is protecting, glutamine is protecting a little less. But when we look at alanine, well, alanine, that's pretty bad, and arginine even looks worse. So based upon these data and some using chamber work, she went on to get a K08 grant. Now, we were very interested in this immune-enhancing diet, and they have different nutrients, but this one we had tested had glutamine and arginine. And so we looked at these data. We said, well, glutamine protects, arginine obviously destroys it. Could it be that it's actually modulating gut IR-induced inflammation? And having the models all set up, we could go in and study that. And so you see this cartoon again. So just how would glutamine protect? And what she demonstrated was that glutamine induces PPAR, which increases AP1 transcription factor, decreases INOS, decreases neutrophil influx and injury. And with arginine, just the opposite happens. And so she had discovered a novel observation of differential induction of AP1 as a way of protecting the gut. So through this, she was able to get an R01 grant, which she is currently studying. Now, true to what Dr. Harkin told us, if you're going to study something, you need to study it in people. So we did a study, a prospective randomized study, where we took patients and randomized them to enteral glutamine at the time they hit the ICU in active shock resuscitation, and we showed that the people who got the early glutamine tolerated our enteral nutrition protocol a lot better than the control. So this is just a proof of concept. So this is the group that I worked with at UT, and it was professionally very satisfying. And I left there in 2006, and people asked me, why'd you leave there? It looks like that was a pretty good deal. Well, they had started a new surgery department, and Barbara Bass, who's a fairly inspirational person, inspired me to develop a new division of surgical critical care and acute care surgery. And for the non-trauma surgeons, this is sort of very important for our future career. If we just stick with the way we're doing trauma right now, there aren't going to be too many people who want to follow in our footsteps. Now, Barbara wanted me to develop a translational research program. And I said, well, I don't know, Barbara. That program at UT, it was kind of like the alignment of the stars. You know, things kind of worked out. And I'm happy to do it, but I don't know what I would study. And my brother Gene had always taught me, well, the way to figure that out is go figure out what type of patients you have and figure out what's killing them and then study that. So it didn't take us long to figure out that it's sepsis. And this is a NSQIP database analysis that we did. And this is on all 370,000 general surgery patients. And if you don't know the NSQIP database, it's actually a very accurate database. And when we looked up the incidence of sepsis, septic shock, pulmonary embolism, and myocardial infarction, that's obviously these occur much more frequently than these, but we spend a lot of time trying to prevent this. And then when you look at the mortality, that's not too bad for sepsis, but septic shock is terrible. 34%, it's as bad as getting an MI. It just occurs much more frequently. And if you sort of add out the numbers, just looking at the number of cases, that's sepsis, that's septic shock, PEMI, and then deaths. We have 2,000 deaths from septic shock and only 193. So we have identified an important problem. We also have identified a confusing problem. There are so many variables that need to be taken into account to understand this entity. And the thing that we realized was that a number of these patients are on different clinical trajectories. And when I was at UT, we had drawn this cartoon, and it was for traumatic shock, but you could interpose sepsis in it. The same thing happens. Some patients come in, and they get septic, and they just flame out. And, you know, that's everybody's seen it. But most patients don't do that. They do a little of this, and then they do a lot of this. So they get a little septic insult, and then they just lay in the ICU. They progressively deteriorate in their immune function. They get sacral decubitus ulcers. They can't heal their wounds. They get infections, and they're out here in this indolent death, and we finally turn off the ventilator and say, let them go to heaven. So if you're trying to do randomized trials and you're entering people that are going this way and going this way, it's kind of difficult to make any sense out of things. And so we propose that the challenge is to predict clinical trajectories. And that's what we want to do, is to understand this. And in the lab, we've shown that if you take alternative treatments that induce protective anti-inflammation, that we can get a more balanced inflammatory response. And if we do that, we won't have very much of that, we won't have very much of that, and we'll have what's called an enhanced preconditioning. And therefore, the people will progress through the ICU and not linger and die. So I have a cartoon. I have a research team. We meet on a weekly basis, and this is multidisciplinary. There's a thing called the Surviving Sepsis Campaign, and I don't know how much you know about it, but it's now in its second rendition. And it's pretty good. It identifies what we should be doing. The inherent flaw is it has 19 recommendations. So if you're the clinician at bedside and somebody's in septic shock and you're supposed to do these 19 things, what are you supposed to do first? And how are you supposed to do that? And how do you make sure you do it the same way every time? Well, that's where this computerized decision support comes in. It actually can apply those guidelines in a very consistent manner. This is just a picture of our paper protocol implementation. We take these gray folders to bedside. We have nurse facilitators that help the team apply these order sets and look at how the algorithms work. And over time, we develop very robust algorithms with algorithms that sit behind them. This is our phase 1 sepsis protocol, which is just for simple sepsis. And then this is phase 2, which is for severe sepsis septic shock. And we actually now have a computer application that we've been running for about 10 months, and it actually works. This just shows you the people that help support it, a bioengineer, an informatics expert, and nurse facilitator. And here you see our critical care fellow, the younger people who use a lot of computers. Just seems like, wow, this is great. They walk up to it and it starts giving them instructions, and they can either say yes or no. If they say no, they simply need to explain why they don't want to do what the computer tells them to do. Now this is kind of interesting. This is Laura Moore. She's one of the intensivists, and she's reviewing the progress through this sepsis protocol with the attending who's off-site, but he's on-site via a robot. So this is Joe Suker. That's his patient. He's not working today, but he's concerned about his patient. So he can sit in his office, beam into the ICU, and then he can go over the sepsis protocol and figure out where we are in the management of this patient and feel good that he hasn't abandoned his patient. Now, as we started doing this, one of the inherent flaws was detecting sepsis early. If you wait until the patient goes into septic shock, you can treat that, but you just converted a 5% mortality into a 35% mortality. So how do you pick this up early? And Laura Moore developed this sepsis screening tool, and this is done twice a day. It's pretty simple. Now it's web-based, and it starts off with a nurse filling out a SIRS score. So, I mean, this really isn't very hard. They just have to collect 4 pieces of data, and then they circle these little numbers, and they get a score. If the score is over 7, well, then they give this nurse practitioner a call and say somebody just screened positive. The nurse practitioner has this worksheet they work through, and they try to figure out what's the likely infection. This person, it appears to be a pneumonia. They then call the intensivist. We review the data, and in this case, I felt the patient had a ventilator-associated pneumonia. And I say, okay, let's start the sepsis protocol. The patient's sick. They're going to go into phase 2, get the computer, and start applying it. So just to wrap up quickly, we have a sepsis database now that we're developing. We're about 270 patients into it. So it's starting to get robust enough that we can start doing epidemiology studies. We're concurrently measuring cytokines, and we should be able to come up with some interesting observations. One of our residents is working in the UT labs, learning these SMAO models. She's looking at how lactoferrin modulates gut IR inflammation. And then Hytham, who was my diamond in the rough, has ended up at Johns Hopkins, and I just recently got him to come back to Houston. So we're starting our lab up, and he's going to be an excellent research partner. And then these are the potential stars. And as I said, the secret to this is to align all these people. There are some very talented people here, and we got to simply capture a couple of them and say, okay, we're going to do some high-end translational research. So my conclusions are, when you talk about translational research, you should focus on an important and confusing problem. With the best available data, you should choose, and your clinical insight, which is very important, you should propose new cartoons and develop win-win hypotheses. You should attract new investigators, develop research partners, gain from their expertise, but more, just as importantly, provide relevance to them. So if you're a basic researcher, having somebody show you what you're doing, how it relates to a patient in an ICU, is tremendously attractive. And then you need to test the cartoon both in the lab and in focused clinical studies. And we really should function as the bridge that goes between the basic scientists and the clinicians at the bedside. So this is a quote from Albert Einstein. Significant problems we face cannot be solved at the same level of thinking we were at when we created them. And then finally, going back to the title, imagination is more important than knowledge. and I thank you for your attention.
Thank you, Fred.